Right, so the first part of today's lecture will be on the final project. I'll go through the problems with you and I'll ask your questions if you have any questions regarding the problems. So for this project, our hope is that it will be a very good way for us to see how people might do analysis in a very different way. So we basically hope to use this class project as a way to assess the diversity in single cell data analysis. So that's why, as I said in the first lecture, so the final project report part is based on completion. It's not based on whether your results are right or wrong. And the results is because it's very normal that you may get different results from your data using your analysis. So therefore, we want to see your subjective judgment in data analysis. How would you do cell type annotation? So here is the PDF you can download from the assignment final project, what I posted already. It has two parts, and each part has different data sets. So for first part, we will focus on the bone marrow, which will give you two data sets. So just as a hint, the two data sets may contain different cell types. So that's why we are giving you this list of cell types as the superset, in the sense that the cell types you would find in each data set may be all of them or maybe only a subset of them. And that is where your judgment will kick in. Another part we need your effort or need your judgment is to find the cell type marker genes. So for each cell type, you would be able to find marker genes on the internet, right, or relevant literature or some database. But in reality, right, if you are working in a lab that asks you to do the cell type annotation, where to find cell type marker genes is your choice. So that's why we leave this open to you. And probably you can start with finding marker genes before you do this, because that will take some time. And you may find different marker genes than others because you may look for different sources, but that's part of the diversity in your analysis. And that's how I want to say why people might annotate the same data differently, depending on their marker genes and the parameters in the pipeline they use. So to make sure that things are comparable or on the same ground, we will use Surat for everyone. First of all, it's something we cover in the class. Second, it's quite popular and well documented. So therefore, in the Excel sheet, we made it very explicit the parameters you can possibly tune. And we hope you can record what you tried and then record your final parameters, the one you settle down to use for your final cell type annotation. So it's the same thing for both projects, for both parts, I should say. Part one, bone marrow. Part two is this one, human pancreatic and the pancreas data set. So they're both from human. So therefore, when you look for genes, you should look for human genes and don't confuse it with another species. That's very important. So we would have you record your parameters, your final annotated cell types. So you will give a cell, every cell a type label in your submitted Seurat object. So basically you will put this in the metadata under cell type. So we give you the example command here. So finally, you will put your cell type labels as Seurat object cell type, which will be part of the Seurat object metadata. So just make sure that you know the Seurat object where the metadata is stored at. And this is something Zhitian will show you on Wednesday. And if you have any questions, please make sure that you ask him during the lecture about anything about the Seurat usage or the command. So basically, what you will do will be similar to what he will show you, but your judgment will be twofold. First, the Seurat parameters will be your judgment. Second, the cell type marker genes, that will be from your literature search and curation. So there's one question that's only for part two, but not for part one. Is that in part two, we told you there are at most 2 possible cell types. These two, the pancreatic polypeptide cells, PP cells, and alpha cells. So you may have one of them, you may not have any of them, or you may have both of them. That's another your judgment. But if you find them, if you find one or both of them, then there's an open-ended question. That is, can you find new marker genes from this data set besides what you use from the literature as the cell type marker genes for annotation? So this is like a discovery question. Will you be able to find new marker genes? You may be able to, you may not be able to. So this is more like an open-ended question. And we want to use this question to see how people's discover new marker genes will be consistent or very different. So that's mostly of the question. So maybe I can go through the questions in part 1. So then we can read the sentences to see if you have any confusion in understanding this text, because if you do, maybe we need to do some, editing for the parts that may not be so clear. Okay, so let me just read it. In this part, you are given 2 UMI count matrices with genes as rows and cells as columns, the 2 file names. Both these data sets come from the human bone marrow tissue from a healthy donor, which is sequenced by 10x, the 5' technology. Each data set may contain some or all cell types in the following list. There are 21 possible cell types in total, which are listed here. However, it is unknown how many and which cell types each of the 2 data sets contain. You need to determine which cell types exist at your discretion. Please use the SURAT pipeline and search for cell type marker genes to manually annotate the cell types for each data set separately. So you will get but for each data set, you will do annotation separately. So it's not like you will provide just one annotation for both data sets, but you will provide cell type labels for all the cells in both data sets. So it's like you do them separately, even though they are both coupled marrow, but don't merge them or borrow any information from them. You may search online for databases or relevant literature for cell type marker genes if needed. Additionally, please answer the following questions and fill out the Excel file with your answers. Then I will go through the Excel file so you can see what it's like and let me know if you have any questions regarding the format. First one, and under the first one, sub-question 1, what functions in SURAT did you use to annotate cell types? Use Y for yes and M for no to indicate this in the Excel sheets. So in the Excel file, there are multiple sheets. So you will fill out the part 1 dataset 1Q1 and part 1 dataset 2Q1 for this question because we're under question 1. Please record the parameter values to try for each function in the Excel sheets, part 1, again, these 2 sheets as well. So you will see the format. It's pretty standard. So it's like you just need to enter yes or no, Y or N in the corresponding field and the parameter value in the corresponding field. Note that Excel sheet lists the parameters you are allowed to change for each function. You could try any alleged values for most parameters, but for a few parameters, you can only try certain values. If you only use the default values without tuning, or you didn't use the function, fill out NA. So we give you the option to decide whether you just stay with the default, or you can skip the function if not used. Q2, second question. What are the parameter values for each function you decided to use for your final cell type annotations? This is the final result. Fill out them in the sheets part 1 dataset 1Q2 and part 1 dataset 2Q2. See hint 1. So we'll give you some hints below. See hint 1. You should choose one way for normalization. If you choose SC transform, then you should not use scale data find variable features. So this is very important to know because the options of using normalized data or use SC transform, they are exclusive. Your final choice will be one or the other. If you use normalized data, then following that, you need to run scale data and you also need to find variable features. But these two were basically included in SC transform already. So if you choose SC transform, then you don't need them. That's the first hint. Question 3, part 1. What cell type marker genes did you use in your attempt to identify each cell type? List the marker genes in the sheet. It's called part 1Q3. Please use ensemble ID as marker gene names. This is important. We want to standardize our result. And you know that gene name can have multiple ways. Like there are multiple ways to call the same gene for historical reasons. So we will use this ensemble ID just as the gene ID. This is what we will give you in the data set, and we would like you to also use ensemble ID in your report in the Excel file. When you report the marker genes you used, you also use ensemble ID. So the format of ensemble ID starts with ENSG and the sequence of numbers. Basically, ENS is for ensemble, G is for gene. So please don't use ENST. That's what I would like to say, because T will stand for transcript. Ensemble has IDs for gene transcript at the mRNA level. So the difference between gene and transcript is that a gene may have multiple transcripts called isoforms that are from the same DNA same gene DNA region, but they have different sequences in mRNA. So that's why each mRNA transcript is given a separate ID. But here we are doing the analysis at the gene level. So that's why please use ENSG. Question 4. What are your final annotation results? This is the thing you will report using by submitting SURAT objects instead of Excel file. So the previous 3 question answers will be in Excel file, while this last one will be SURAT objects. So you will submit 2 SURAT objects as you will get 2 count matrices. So you will convert the count matrix, each count matrix to a SURAT object to start the whole thing. So please note that this is a RDS, it's an R file. If you read it in using the read RDS function, you will get a count matrix. And you start from there, you convert to SURAT object. So finally, in the question 4, you will submit 2 SURAT objects and you will put cell types you annotated in. So this is for reporting your cell types. Yeah, so that's part 1. Yeah, so the hints are to help avoid confusion. Normalization, one or the other, assay transform or normalized data, the gene ID. Yeah, these are the 2 things we want to clarify. Yeah, so before I talk about part 2, do you have any questions for part 1 or any confusion? Let me clarify why this is the task we gave you. The reason is that if you ask many biologists in UCLA, for example, when would you generate single cell data? Most of the answer would be they want to annotate cell types. They want to discover cell types in their tissue, in the tissue they study, hopefully new cell types. And also they want to discover new marker genes. So basically single cell data is generated as an exploratory resource. People want to know more about our biological system. So that's why we give you this task. So for part 1, we didn't ask you for additional marker genes. Okay, please note that we just asked for your cell type labels. But for part 2, it's a different data set. The number of possible cell types is smaller, right? Previously you have 21. Now you have 2. You need to decide whether they're there or not. So the first 4 questions are the same. So I will skip them. The last question specifically for part 2 is that if you find these 2 cell types, one of them or both of them exist, can you find new marker genes? So this is some exploratory question for part 2. And I want to emphasize that as this project is for completion based, I really hope everyone, or that's the requirement, to do it independently instead of using other people's answers. First of all, the data sets you get may not be the same. So therefore, other people's answers may not be directly the right one for your data set. And second, we want to see your judgment. So therefore, if we spot that, okay, 2 students' results are copying each other, then this will be a plagiarism issue, which will be serious. But if you try your effort but the results may or may not be the expected one, that's totally fine. So basically I want to emphasize that we want to see your independent work. That's the most important thing. And do not worry about the accuracy. Just use your judgment as if you're doing a real research project. Yeah. So that's what I want to say for this PDF file. Then let's move on to the Excel sheet and we can see what it is like. So as I said that you may have different data sets. So basically you will expect to receive an email from Zhiqian today. He will send data set to each of you by email. If you don't receive the email by the end of today with the data set, then please let me know. And also, I want to give you a preview of what we hope to do with the statistics. So we are requesting an IRB approval from UCLA. So once it's approved, IRB is anything if you want to do a study with human. So once we get it approved, then we will send a consent form to each of you to request your consent if we could use your submitted result anonymously. So we will not keep your real name for our analysis. So we want to assess the diversity of the data analysis for the single cell data in the single cell field. So of course, if you don't want to give us the permission to use your data, that's totally fine, but we want to give you the choice. So basically, we hope that we will send you the consent form in the next few weeks, of course, before you submit your final project results. Since the final project won't be due until June the 16th, we still have some time. So we'll send you that form as follows. It's under assignment, final project. I don't know if everyone see that. If you go to Brunner final project, you should be able to see the PDF. No? Let me see where it is. Interesting. Where did it go? Assignments. I see, because it wasn't made public. Sorry, my fault. Okay, now it's published and you should be able to see it now if you refresh the page. Okay. Now let's thank you for pointing out this. Now let's look at Microsoft Excel. So you see, as we said, your answers will go into this Excel sheet. All right, so for part 1 data set Q1, this is for showing your attempts. All right, so basically what we said here is that if you use QC, that's what we introduced at the beginning of the class. Did you filter out some cells? Basically, if you do that, then you will put yes in this field. Otherwise, if you skip the step, you put no here. And for normalized data, if you use this function, you put yes here. And because this is for the attempts, right? You can try different options. So if you tried both normalized data and SC transform, you can put yes on both. This is what you can do. So just whenever you try, you put it here. And for the values of the scale factor, so basically we said that in this case, you could only try these two values. We only give you the two options, 10K or a million, not other things. But if there are no specification, for example, variable feature number for SC transform, or here, number of features, n features for fine variable features, we give you the flexibility. Remember that the default is 2,000 here, but you can go with down to 1,000 or go up to 3,000. So if you just change this parameter, put your values here. And we ask you to separate the values you tried by this semicolon. So you just say, if I tried 1,000, 2,000, 3,000, I just put 1,000 semicolon, 2,000 and 3,000. So it's this format here, we give it down here. And if you just use the default value, say 2,000, no tuning, just put NA here. And for the scale data, just say you use it or not, because there are no parameter to tune. And for PCA, use it or not, right? We're here for our lecture. You put your number of pieces here. Then In the following lectures, we will talk about find neighbors, find cluster, or run UMAP or t-SNE, and find markers. So even though we haven't talked about the specifics there yet, Zhitian will show you his pipeline tomorrow, just as a preview. And there are parameters you could tune for each. So this is part of the why Seurat data analysis, when different people do it, you may end up using different things. And so for most people, users, maybe they will start with defaults and then see if the results are reasonable and then go back and change. That's the reality. That's what most people do. But how you will do the analysis, you will give it a try. And especially if we give you the total of 21 cell types, you kind of have a target in your mind, right? So that's kind of the direction you're guided to go to. But please be aware that not all cell types must exist in your data. So that leaves the room for your judgment. Okay, that's for dataset 1 Q1, dataset 2 Q1, same format because it's the same question, just two datasets. Then dataset 1 Q2. This is your final result. So the final parameters you choose, you decide to use. So for that, you need to make sure the results are, the parameters are unique. The most important thing is that you would not use SC transform together with normalized data. So for example, if you choose yes for SC transform, then for normalized data, it will be no, scale data will be no, find variable features will be no, because these were all included SC transform. That's very important to make sure that your results are self-consistent. And in this case, every field will only have one value instead of values separated by semicolon. And the same thing for dataset 2 Q2. And for Q3, here, this is the thing I want to emphasize. We want you to list the marker genes you use. And of course, different people may end up using different marker genes. That's normal. But to help us process your results easily, we hope you can enter one gene for one Excel field. So like this is for this one cell type. If you have 3 marker genes, then you have here, here, here. So you just put them one by one. Okay, if you have 4 marker genes, you put it here. So that's why we give you one example. The example is showing you that the gene will be in the ENSG format, and they will go one by one, horizontally. So if you have all those cell types, you put them here. So, and also this is the marker gene you should have before you do this whole Seurat data annotation. The reason is that this is how you would rely on to annotate your cells. So that's why this information should be there before you start your analysis. Okay, that's all for part 1. Only the questions 1 to 3 are going to be answered by filling out Excel file. Okay, so your final project submission, let me just clarify again. It's this Excel file and 3 Seurat objects corresponding to the 3 Seurat, corresponding to the 3 count matrices we gave you. For part 2, I will skip part 2 Q1, same thing. You will fill out your tried values. And here you will fill out your final parameter values and your marker genes for the 2 cell types. And this is for the new marker genes you potentially discover from the data set. So that's it. So besides this Excel sheet and the 3 Seurat objects, you will each do a final 5-minute final presentation in the last week. That's also part of the plan. So basically this Excel sheet and the final Seurat objects will be due later than the final presentations. So, but for the final presentation, it's just trying to let you show some of your results. Yeah, and also you can see what other people find. But as I told you, your data sets may be different. So therefore, other people's results may not be so relevant, but we'll see. Yeah, so basically, if you have any questions or concerns, just let me know in the meantime, because this is like our first time doing this, and we can adjust our plan accordingly if you have any concerns or suggestions. So we're very open for your input. Okay. Any questions or concerns or suggest, or I would say, yeah, anything you want to say before we move on with our lecture materials? All right, so if there are no questions, you can let me know later if you have any questions coming up. So then we will continue with our lectures. And the next homework will be assigned this week, which will be mostly focusing on PCA and dimension reduction we recently covered. So last time, if as a very brief overview with what we talked about, last lecture we talked about choosing the number of PCs using the elbow plot, and needle algorithm is an automatic way to help you choose it. But of course, you can use your eyes to see where you think is the elbow. And there are other ways which I didn't list here, but you could actually use. So you're not required to just use needle. You can try other things. So for example, maybe I can add it here. Yeah, I should do it. So yeah, so determine the dimensionality, right? So if I go back to here, so maybe I'll just quickly put it here in the lecture slides. So you could use eyeballing, and you could use the needle algorithm. And in the R package, there is a R package called FindPC, which is a recent work published in bioinformatics. So you can also check out this package as an alternative way, so to help you decide. But the interesting thing is that R package FindPC doesn't include the needle algorithm. So yeah, which also says that choosing the dimensionality is something subjective, because we are not handling a supervised learning prediction problem, but an unsupervised learning dimension reduction problem. So I remember I said this in the first lecture, right? For unsupervised learning, you only have a matrix. In our case, the matrix has either cells as rows, genes as columns, or cells as columns, genes as rows. But basically, it's a cell by gene matrix. And you don't have the cell type labels as an additional Y vector for you to predict. So many algorithms that will give you a Y hat for prediction, they belong to the supervised learning category. But everything we said so far is only for X, the matrix only. So you don't have a Y, and it's an unsupervised learning setting. So then last time we said that, okay, for the GLM-PCA, we finished PCA and we started talking about GLM-PCA, which wants to work on the count matrix, the UMI counts directly. I should go to full screen. The UMI counts directly instead of working on transformed and scale data as normally distributed data. So it wants to avoid that normalization and scaling step we did in the default Sura pipeline. So first of all, this is what we already said last time. So the author's motivation is that the log 1 PCPM, right? Even though we now Sura defaults log 1 PCP 10K, but you will still have a similar thing, maybe not as severe as here, that you will have a bigger separation between the counts that were zeros and the non-zero counts. So you make them into very big two modes. This in statistics is called a bimodal distribution. You have a mode here, you have a mode here. So a mode is a maximum in density. But the original data may just be like this. It could be fit by a negative binomial count distribution, just one mode. but the data transformation will create the two modes. So this is the motivation and concern of this GLM-PCA authors. That's their motivation. So I said, I think this is where we stopped last time. So another look at PCA. Recall that we spent quite some time on PCA because I think it's very important. We talked about two ways before this one. One is that we find the first PC to maximize the variance of your cells in that PC. So maximize the PC's variance of PC scores in PC1. Second, we want to have the best reconstruction from the PC, from the low dimensions to the high dimensions. And PC represents the best low dimensional reconstruction. So the idea is that if you have originally P features, your P genes, your cells are in the P dimensional space, we find K PCs to represent the cells. And then if we want to go back to the P original features from the KPCs, we could do the reconstruction. And recall that we just multiply the K by P matrix, which is the transpose of the P by K matrix you obtain from the PC loadings, right? So that is the projection. You project P dimensions to K PCs using the P by K matrix. And then if we take the transpose of that matrix to become a K by P matrix and multiply this matrix to the cell by KPC matrix, the P dimension reduction, then you can restore the original P dimensions. So here is the third way. The third way is that you're still talking about the representation, but kind of very related to the reconstruction I just said. But here the notation is written element-wise. So basically, UI is if we think about Zij for cell I in gene J, right? Then we will call UI as the PC scores of cell I in the KPCs, and Vj as gene J's loadings in the KPCs. So then we can do this vector inner product. So this UI and Vj are both k-dimensional, corresponding to the KPCs. Their inner product is just a scalar. And we hope the scalar to be as close as possible to Zij. And this perspective is equivalent to that reconstruction perspective. So this one says minimizing MSE is equivalent to minimizing the Euclidean distance metric between the embedding and the data. So this is the embedding and this is the data. And minimizing this can be also interpreted equivalent to maximizing the likelihood of a Gaussian model. So if you believe that Zij is following Gaussian distribution, where the mean is just a low dimensional representation, this UI transpose Vj. If you believe this is the true mean subject to some Gaussian random error, then this is, you can write down a likelihood based on the Gaussian model, and maximizing that likelihood becomes just minimizing this MSE, mean squared error. Okay, this is the PCA perspective. Okay, then I want to make a connection to GLM because it's used in the name, generalized linear model PCA. So then we need to first start talking about the linear model. So the linear model LM has this Gaussian model assumption where the Gaussian assumption is applied to the random error term. So basically, we assume that for each observation I and J, observation I, observation J. So in our setting, we interpret our cells as observations. We interpret our genes as our features or variables. So forget about the linear relationship, and so basically don't try to so in this case, the linear model is introduced, I would say, outside of single cell data analysis for now, or just introduced in a general form. Many of you might have heard or learned this before. So here, the interpretation I want to add, which is to connect with this normal Gaussian model, is that we will interpret this linear model as a Gaussian linear model. So in the setting you're familiar with, the error terms are IID, normal, same mean, same variance, and yi depends on this linear function. And we call this simple linear model because there's only one predictor, one feature. We want to see how xy can predict yi. We assume that yi is a linear function of xi plus error term. Okay, and this is the vector form I remember we also introduced last time. Stacking things together, y1 to yn, the n observations become an n by 1 vector, and we put the predictor values also in the column, but we also add a column once to represent the intercept we allow, the 1 beta 0. And then the parameter vector is stacked into a two-dimensional vector beta 0, beta 1. And the n error terms are stacked into an epsilon vector. So therefore, the model becomes written in the matrix form. The model becomes y equals x times beta plus epsilon. The dimensions for each term is n by 1. And following that, we denote the row vector, each row of the design matrix X, we call this design matrix as Xi transpose. So this allows us to generalize. So with the design matrix written as this, Xi transpose, X1 transpose, X2 transpose, Xn transpose, and by P matrix, we can write the linear model as a two-layer structure. This will help us understand generalized linear model. So there's a systematic structure that specifies the relationship between the mean of yi, we call mu I, and xi transpose beta. So the linear function is here, linear, right? Xi transpose beta is linear in the predictor. And it's about a mean of yi. There's no randomness. It's a fixed structure. The random structure is in the second layer, which specifies the randomness in my response yi. Why it is different from the mu I? What makes it different from mu I? So for this, yeah, like I said last time, I should pen color. Yeah, so this one should be crossed out. Yeah, so the matrices should be crossed out. It's just yi should follow independently with their own mean specified by the systematic structure and the common sigma square for the common variance. And the result, the reason why we have this is because we have the error term. We have the error term from this normal distribution. So it should be I here because epsilon I follows this normal distribution. Oh, sorry, maybe my bad. I should erase it. Let me see, eraser, laser pointer. No, I don't know why it's not working on my computer. Automatically pointer. Okay, never mind. So basically, I'll make the correction later in the slides. So what I want to say here is that so this normal distribution for yi is derived from the normal distribution for the error term. So we have a mean structure and we have a distribution. So then when we move on from the linear model to the generalized linear model, we change the normal distribution for yi to another distribution. So logistic regression as the most commonly used generalized linear model, here the yi is not continuous. So to have this yi follow a normal distribution, as you know, the normal distribution can take any real values. The yi must take continuous values with digits, real values with digits. But here, the yi is specified to be just 0 and 1. That's a response. Whether you vote yes or no, in this case, it's not reasonable to assume that yi follows a normal distribution anymore. So therefore, a common way is to assume that the yi follows a Bernoulli distribution, because the Bernoulli distribution is exactly that give you 0 and 1s. So there's only one parameter in the Bernoulli distribution, pi I. Pi I denotes the probability of getting a 1. And for Bernoulli yi, we know its mean is yi, and we know its variance is just pi I times 1 minus pi I. And pi I is a probability of success is between 0 and 1. So this is a value between 0 and 1. So one thing we need to note here, different from the linear regression, is that the variance of yi changes with pi I. So here the variance changes with the mean. While in the normal case, right, linear model case, what's the variance? The variance is just sigma squared. And sigma squared doesn't depend on mu I. So they are basically decoupled. They don't depend on each other. But here the variance depends on the mean. Recall your first homework. We said that this is called variance not stabilized, because for variance stabilization, we want the variance to be unchanged while the mean may change. That's the property we want to be Gaussian-like, because for Gaussian, we do have this property, right? So sigma squared doesn't change with mu I. But in this case, for Bernoulli random variable, we wouldn't have that. Naturally, the variance depends on the mean. Then the question is, how do we link expectation of yi to predictors? Just like the systematic structure here, we want to link mu I to xi transpose beta. But how could we do this with yi, with this pi I, right? Because pi I is between 0 and 1. How do we link it to predictors? So if we just use the same systematic structure as the linear model, we call this proposal 1, then we basically assume that pi I is just xi transpose beta, right? So this is explanation for what is xi transpose. It's a p-dimensional row vector containing the predictors values. Yeah, so basically the first one is just a constant for intercept, and beta is the p-dimensional column vector containing the coefficients. However, we run into a problem if we want to use this proposal 1. Pi I has a range from 0 to 1, while xi transpose beta has a range from negative infinity to positive infinity. It depends on what are the predictors. So if in this example, your predictor include age, whose range is definitely not constrained to 0 to 1. So it's very possible that if your predictor's value range is not 0 to 1, there's no way for you to, it's not easy to constrain that the right-hand side xi transpose beta, even though you're going to find beta, but this value is very hard to be constrained to 0 to 1. So therefore you have a mismatch, right? Left-hand side 0 to 1, right-hand side negative infinity to positive infinity. So it doesn't make sense. So then another idea is that what if we define it to be an odds ratio? So this is called an odds ratio, which is commonly used in many biostatistics problems. Just be aware. So if we do pi I divided by 1 minus pi I, this is the odds ratio, right? It's basically odds of getting 1, odds getting 0, and their ratio. So it's the probability of y equals 1 over probability y equals 0, xi transpose beta. This is proposal 2. And however, the range is still not matched yet. So here, what's the range of the left one? It's from 0 to positive infinity. When does it get 0? When pi I is 0, you get 0. When does it get positive infinity? When pi gets 1, you get 1 over 0, you get positive infinity. So left range is 0 to positive infinity, but the right range is negative infinity to positive infinity. So you miss the negative part. Then we do another modification, which is our final proposal. That is, we apply log to this ratio. So when we apply log to this ratio, log 0 becomes negative infinity. Log positive infinity stays as positive infinity. So we could get the whole range. And this range matches the range of xi transpose beta. And that's the proposal 3. And this log odds ratio is given, this function is given a name as the logit function. Logit function, logit pi I, is basically defined as log of pi I over 1 minus pi I. And then the systematic structure for logistic regression is logit pi I is xi transpose beta. And again, there's no randomness involved in the systematic structure. Everything is fixed. The randomness of pi I, of yi, in logistic regression comes from Bernoulli. That is, if you have two individuals whose predictors perfectly match, like they are same age, same gender, same education level, but their voting outcome may still be different. Like one person may vote yes, another person may vote no, and that is due to the Bernoulli randomness. So that is the fact that if your pi I is far away from 0 or 1, so if it's 0.5, that's where the biggest randomness happen. Because for 0.5 Bernoulli probability, you are just doing a fair coin toss, right? You have equal chance of getting a 1 or a 0. Even though two people share the same pi I as 0.5, they may get different outcome. So the randomness is from Bernoulli. That's the most important thing I want everybody to know. So, and the Bernoulli replaces Gaussian in the linear model. Okay, so this is just showing what the logit function looks like. So first of all, we are plotting pi I in the x-axis, logit pi I in the y-axis. Then logit pi I is a monotone increasing function. Whenever it's negative here, then the odds is less than 1/2, right? So basically for pi I less than 1/2, your logit pi is negative. For pi I greater than 1/2, logit pi is positive. Yeah, and beta j, which is the jth element of beta. Here, beta j is the coefficient, or we can say slope of the jth predictor. Then beta j measures the increase in logit pi I when xij, when the jth predictor value of observation I, we call this xij, increase by 1. So if this xi's jth predictor's value increased by 1 in the given unit, then beta j measures how much is increased in the chance, but the chance is transformed into the logit. So you see, the monotonicity is nice because we still say, okay, the larger the probability pi I, the larger the logit pi I. However, it's not monotone, so we basically convert range 0 to 1 to this whole real line from negative infinity to positive infinity. Does it make sense too? Okay, that's the logit thing. Then we give you another example of GLM, Poisson regression. Now your yi are counts. They're not just 0, 1, but they can go beyond 1. You can have 0, 1, 2, up to infinity, possibly. So if we assume the count for each yi is following a Poisson distribution. This is the so-called probability mass function, the probability yi, big Y, peaks value, small yi. And for Poisson with one mean parameter mu I, then yi's expectation and variance are both equal to mu I. That's our mean of yi. And the systematic function, systematic structure again tries to link the mean of yi, mu I, to the predictors. So previously we have this logit pi I. So here pi I is the mean. So you can write pi as mu I as well if you stick with mu I being the mean of yi. So logit links this mean of yi to the linear predictors. So that's why we give this logit function a name. We call it a link function. That's something I didn't emphasize yet. Link function maps the mean of yi to the linear predictors. Here is log function. And just ignore the jargon because we didn't go into that level of detail, canonical link function. If you are interested in this and learning more about this, I'm covering this in my other class. I'm teaching right now the 205 class, Stat 205, which is specifically about generalized linear models and beyond. So you're very welcome to check out the video recording for that when we talk about canonical link function. But just think it from a simple way. For Poisson, mu I is non-negative. So if you just have mu I equals xi transpose beta as the Gaussian linear model case, you will still have a mismatch of ranges. Mu I is non-negative. Xi transpose beta can be negative. They don't match each other. So therefore, we use the log to convert mu I to the whole real line. Because when mu I is between 0 and 1, log mu I is negative, right? Log 0 is negative infinity, and log positive infinity stays as positive infinity. So log is also a monotone function. It maps the positive range of our real line to the whole real line. So then log mu I is real value, which matches the range of Xi transpose beta. So in this case, the effect beta j, jth predictor slope, is effect of jth predicts 1 unit increment on log mu I. Then if you take exponential transformation of beta j, it's the jth predictor's 1 unit increment on mu I. So it's a way to interpret what the slope means in the Poisson linear model. Okay, so as you can see that for different types of generalized linear model, what we change is the random structure because the response yi would follow a different distribution depending on whether it's real values, variable, whether it takes real values or whether it takes binary values or it takes count values. So depending on each case, we may assume a different distribution to be reasonable. And correspondingly, once you have the random structure, you have the mean of yi, which is mu I, and you always want to link mu I to your linear predictors. So how do you do it? You may need a different transformation depending on the distribution because the mu I may have different ranges. If it's just between 0 and 1, we use logit. If it's non-negative, we use log. So these are the most common ones. And if it's Gaussian mu I can itself take real values, then we don't do any transformation. It's just mu I equals Xi transpose beta. All right, do we have any questions before I continue? Just the GLM part, generalized linear model. And G stands for generalized, which means that we go beyond Gaussian to other distributions. Okay, now GLM-PCA. So recall that, okay, let me scroll a few slides back to connect it. This is what is assumed for the normalized data. Normalized data from single cell data, Zij, cell I, gene J. Because it's normalized, we assume it's Gaussian distributed, and we do PCA on Z, Zij, to get the mu I transpose, which is for cell I's PC scores, and Vj, which is for gene J's loadings in the KPCs. So that's what we had for PCA. Then let's move to GLM-PCA. So here, if we replace Gaussian with Poisson, which approximates the multinomial, I remember I mentioned this in the previous lecture, but if there's something not still new to you, I suggest a review of this topic. So, or search for Poisson approximates multinomial, which is very good to know. And so that justifies why Poisson is so widely used for sequencing data, because sequencing experiment is essentially multinomial. We have a total number of reads to be assigned to different genes in a multinomial way. So then for Poisson, here, the authors consider Yij, which is the UMI count of gene I in cell J, follows a Poisson distribution, and Ni is cell I's library size, total UMI count. And then multiply something which can represent the proportion, or what I should say, which represents the true expression level of gene J in cell I. Okay, and then they said that the true expression level, because it's non-negative, right? Then we assume the log of the true expression is mu I transpose Vi. So just like here, log of true value is the mean. Log of the true value is the linear predictor. It's the linear predictor here. And here we have the log of the true value, which becomes mu I transpose VI, because this exponential term is to represent the true value. So again, the idea is that same as the Gaussian. In the Gaussian case, we have Zij follows normal with mean Ui transpose Vi. So we believe that this low rank representation is the true value in the Gaussian case. But here, this low rank representation may take real values that can be negative. Therefore, we take exponential transformation to make it non-negative, so they can serve as a valid true value. So the reason I mentioned GLM is that I think that's essential for you to understand why we have an exponential term here. This is because of the log term here. It's one way or the other. So basically, you move, if you do exponential transformation on both sides, you get mu I equals to exponential xi transpose beta, and that's what we have exponential here. And can parameters of Poisson distribution be zero? Yes, it can be zero. Yeah. So when you have, let me try to move away from the laser pointer. Yes, it can be zero. So if it's Poisson zero, then you just have a point mass at zero. So zero counts only. Yeah. Or alternatively, in the case of overdispersion means that Poisson requires mean and variance to be equal, but when you have overdispersion, the variance can be bigger than the mean, right? Then we may approximate the Dirichlet multinomial using a negative binomial likelihood. So negative binomial is the overdispersion version of Poisson. This is something we also introduced last time in a few lectures back. Because of the existence of the dispersion parameter of the negative binomial's variance is bigger than the mean. So you just include this dispersion parameter for gene J. So you make the distribution gene specific, which is something also I agree with, because we consider cells to be observations from some distribution, while genes are variables. So you have a gene specific dispersion, and this mean is the same mean as before. And so in this paper, it says that the GLM-PCA was originally proposed in this machine learning conference. Some of you may know, now it's called NERPS, used to be called NIPS, and it started in early 2000, or maybe even slightly before that, late 90s. So then actually they published papers in the NIPS conference. And this was a then NIPS paper, a generalization of the principal component analysis to the exponential family. Yeah, so basically with this, we are going to fit this model, becomes an optimization problem to maximize the likelihood. By maximizing the likelihood, you are going to search for what is UI vector k-dimensional, what is VJ vector k-dimensional. So of course, the optimization problem will be more complicated than PCA, because PCA is just eigen decomposition. So this one, you need to have some way to turn this into an eigen decomposition problem in your iteration. Basically, I can tell you it's an iterative algorithm. So then you end up getting UI and VJ. So the results are still the same format as PCA, but you do it by maximizing this likelihood. Okay, I hope you get a high level idea of what is going on here. We save the effort of data transformation, right? But we put the effort into optimization. So you must pay the effort one way or the other, but yeah, and they are parallel approaches. Transform your data to fit a normal distribution, Gaussian distribution, or keep your data, but propose a more reasonable distribution. But the maximum likelihood estimation problem may become harder. Okay, so as an illustration, consider GLM-PCA with a Poisson approximation to a multinomial likelihood. The objective function to be minimized is simply the overall deviance. So this is something I will probably just quickly talk about, because minimizing the deviance is equivalent to maximizing the likelihood. If you understand maximizing the likelihood, what that means, then you would directly get the minimizing deviance. Deviance, by the name, is how your data is deviated from the model, or the model's deviated from the data. That's the discrepancy you want to minimize as a way to fit the model. So if you minimize the deviance, you find the best parameter values of your model to describe your data. So the deviance is going to replace what we have as the mean squared error, MSE, in the Gaussian linear model, or Gaussian, or the PCA, Gaussian PCA. So basically, MSE is the special case of deviance when the distribution is Gaussian. That's basically it. But deviance is more general. You remember in the Gaussian case, we have the minimizing MSE equivalent to maximizing likelihood. Same thing here. Minimizing deviance equivalent to maximizing likelihood. And here, if you look at the log mu ij, and mu ij is basically this, log mu ij mean can be written as this. Yeah, so I'm just going to skip everything. But basically, I think there's like a change of notation because this SI is the NI, and these terms are just this spelled out. You basically have this UI transpose Vj spelled out in this way, in the scalar form. Yeah, so SI is NI, and optimization proceeds by taking derivatives with respect to the unknown parameters. Vj1 is the gene-specific intercept, and the remaining are the latent factors. So just to be interpreted as PCs. All right, so GLM-PCA is you can consider to be the Poisson version or negative binomial version of PCA. And as I said, the advantage of doing that is you don't need data transformation, which can be arbitrary normalization. But what the price you pay is longer computational time. But the good thing is that recently, after this paper, there has been faster versions of GLM-PCA proposed. So in your homework, next homework, which we will assign, we hope that you can try to explore the fast version and see how it performs. Okay, so finally, I think this is something I want to share with you, and it's very good to know, multidimensional scaling, MDS. MDS is closely related to PCA, but your input data is different. So for PCA, you will input N observations and their P features, right? Like N cells with P genes, that's your input. But MDS can start from just an N by N distance matrix. So you may not have the original data for the P features, but you may have the pairwise distances of the observations. And this could be the case for some data sets. For example, if you have a network data where the observations are the nodes in the network, such as a friendship network, you know how good we are, how close we are as friends, which can be encoded as distances. If we want to visualize ourselves in a 2D map, so we can see this pattern, see the distances, that's the MDS. So the hope is that for each observation, we want to find the two-dimensional coordinate or two-dimensional embedding so that the pairwise Euclidean distances the observations have in the 2 dimensions would resemble what we input. We resemble the N by N distance matrix. Okay, so that's the thing. And this is the Wikipedia introduction. And this is showing an MDS embedding of voting patterns of the House of Representatives of the US. Right, so each dot is a Republican, it's a member. So they're colored by Republican and Democrat. But you can basically see that similar people are indeed closer to each other in the 2D visualization. So here is the technical part, but I'll try to be clear to help you understand. Let me try to, I think this is something I need to, N times P. Yeah, this should be done. So how is this related to PCA? We will use eigen decomposition for both. That's the similarity. So given pairwise similarities, so distances can be converted into similarities. For example, if the similarity is Pearson correlation, absolute value Pearson correlation, right? 1 is similar, 0 not similar. We can do 1 minus Pearson correlation to make it distance. So there are different ways to convert similarity to distances. One is larger, the other is small. So we want to find a map to preserve the distances, to find the low dimensional space for y1 to yn, and each one is for one observation. Our input is an n by n distance matrix, capital D matrix, Dij, n by n, so it's symmetric. We don't actually require to be a metric, so it may not, by metric, metric has some very formal mathematical definition to be a metric. It doesn't have to be. But just for simplicity, you can consider to be a distance metric. It doesn't really matter for the following discussion. And the output is y1 to yn, and you need to specify the p typically as 2 for plotting purposes. Your hope is that this is denoted by yi minus yj double bar 2. So mathematically, this means the Euclidean distance. And some people refer to this as the L2 distance, the same thing. So you want this Euclidean distance between y and yj to be very close to Dij in the input. And this is the definition of Euclidean distance just for your review. Let's say we have p dimensions, we let k go from 1 to p. In every dimension, we take the square distance between y and yj, and we sum over the square distances, take the sum, and then we take the square root. That's the definition of Euclidean distance. But if we take this matrix form and expand it, actually it can be written, and yi, yj are both p-dimensional. It can be written as yi transpose yi plus yj transpose yj minus 2 times yi transpose yj. So the reason we write this matrix form is that then we realize to find the yi and yj, we can transform the problem into finding such a matrix B. B is defined as YY transpose. What is dimension of Y? Y, we stack the YI's by row. It's N by P matrix. N observations, the P dimensions, and here P is 2, so it's N by 2. And then YY transpose is an N by N matrix. And the reason we want this is because doing this B construction, it's IJ's entry Bij. It's basically Yi transpose Yj. That's what we need in this composition. So essentially, using this B matrix, we can write this Yi and Yj Euclidean distance square here as Bii plus Bjj minus 2 times Bij. So then if we can just find the B matrix, we can find the Y1 to Yn. That's how we transform the problem. Okay, so that's what I said, right? So if we can find this B matrix, then Yi minus Yj squared can be written as Bii plus Bjj minus Bij. And if our goal is to set this is our ideal goal. The Euclidean distances of y and yj is equal to the input Dij. Then we can say, okay, we can set this left-hand side as our goal, which is Dij squared. Then we have this thing, Dij squared equals the right-hand side. And we know all the Dij's, so we hope to find the B. And finding the B, after that, we use this definition of B as YY transpose, decompose B to find Y. So that's the whole idea of doing the MDS. So going from D matrix to B matrix and going from B matrix to the Y matrix. But there's an identifiability we need to consider. Since Yi, we can shift each Y by the same constant vector mu. Then you see this shifting will not change the Euclidean distance of Y and Yj, but it will give us different embeddings. So this is called unidentifiability. You can have 2 or more than 2 sets of Y1 to Yn that will give you the same Euclidean distances. To make sure we have identifiability, we add a constraint. Here's the constraint we are familiar with, centering. We make sure that for each of the new dimension, we have 2 new dimensions. For each new dimension, the yi's sum up to 0. So all the n observations are centered in each new dimension. So every dimension yi is centered at 0. So with this, we solve this identifiability issue. Okay, so here is a bunch of math just to help us understand what's going on here, which I think is crucial if you really want to understand the MDS. So we can come up with 4 equations to help us solve B from D. First of all, because we have the centering I just introduced, it will make things much simpler for us. Because we have that for each k, summation of I from 1 to n is 0. Because of this, we can actually show that the summation for Bij, the B matrix, if we fix J, sum I from 1 to n is 0. And because of this, then if we look at this sum of Dij squared, left-hand side, we take the sum for I. We can show that it's equal to the trace of B. Trace is the sum of diagonal elements in the B matrix and this n times Pjj. So I skip all the derivation here, but just to show you that this is actually pretty natural. If we take the summation of I, then the last term, summation of I of Bij will be 0 based on equation 1. And sum of I of Bii is just the trace, right? And sum of I for Bjj, because Bjj doesn't involve I, so it's repeating n times Bjj. So it's straightforward. And then if we take the summation over J instead, by symmetry, we will get n times Bii and trace B. Finally, if we take the double sum of Dij squared, then we will be able to get 2n times trace B. So these 4 equations allow us to solve Bij based on the D's. So it's basically Dij squared minus the average across I minus the average across J. And I think this should be plus. I will double check. I think this should be plus and plus the double average of D. So basically with this, we can actually get Bij. So this is also something very useful to know. The B matrix actually has a name. It's called the gram matrix. It has a name, gram matrix. And we know that we can always turn a distance matrix into a gram matrix in this way. So I would say this is a general result. It's not a specific result for MDS. But then with a gram matrix, because B is defined as Y transpose, to find Y, we just do eigen decomposition, which is very nice. So for now, B is not necessarily low rank, because if we set, if we set this equation here to be exactly equal, right? We want the representation by B for Yj Euclidean distance to be exactly equal to Dij, then mathematically, if we know Dij is the Euclidean distance of I and J in the original p-dimensional space, and if it's full rank p dimensions, then we would need B to have rank p to be totally equal. So you can achieve this equal sign. Otherwise, it wouldn't be equal. But we only want Y and Yj to be 2-dimensional. Then what do? We need to find the low dimension, 2-dimensional approximation of B. So that's why when we do the eigen decomposition, it's natural. So basically, this lambda matrix is the eigenvalues, and V and V transpose are the eigenvectors. It's natural just take a square root for each eigenvalue. So we put them into the front and the back. So we have 1/2 here, 1/2 here, and we define that's the Y. So you can see that if you define Y in this way, then Y times Y transpose is naturally B. But this Y may not be just 2-dimensional. To make it 2-dimensional, we do the truncation on the eigenvalue. This is the very typical strategy for truncation, for approximation. And you just keep the first 2 eigenvalues, set the rest to 0. Okay, and basically you just take the first 2 eigenvectors, first 2 eigenvalues, take the square root of the first 2 eigenvalues, and that is your Y. So then you'll get a 2-dimensional embedding. And you can see here the similarity or connection to PCA is what? Eigen decomposition. So here we're doing eigen decomposition on the gram matrix. For PCA, you are doing that. We did eigen decomposition on the covariance matrix, X transpose X. But the idea is very similar. So that's why I'm saying that this is called a classical MDS. It has very strong similarity with PCA. We're both working with linear algebra to do dimension reduction, and the strategy is both eigen decomposition and truncation for top eigenvalues. These are the same, but the input is different. If you ignore the technical jargon or technical math details, I include here just in case some of you are interested. But I think the key thing here is that input is different. Here you get an input of a distance matrix. Well, for PCA, you don't get an input of a distance matrix, but N by P original matrix. However, there's an equivalence. If you construct Euclidean distances of your observations using the original P features, call that as the distance matrix. Do this MDS. Then the embeddings you get here is essentially the top two PCs you get from PCA. So they are equivalent. All right, so this is something I would hope you can try in your next homework. So basically, I will make the problem explicit, so you can try to find the equivalence between PC and MDS. Okay, so that's all I have today. And if you have any questions, you can ask them on Zoom. Okay, and as an announcement, the Wednesday lecture will be a tutorial, a demo of the cell type annotation you will do in your final project. Is that online? I haven't, so I'll discuss with Zhitian, but I think Wednesday, I think the default is that this class is in person. And I will ask him to record. But if you guys have no issue with doing it online, or do you have a preference to do it online or in person? Because I think we default the classes in person, but we can do the recording anyway. Okay, so then that's all for today. Thank you. So then I will see you on next Monday. Thank you.